Hi everyone, myself P.ANUHYA studying B.PHARMACY first year in Maharaja's College of Pharmacy. Today I am going to discuss about the topic de novo synthesis of fatty acid. Let's now go into the topic. Here the dietary carbohydrates and amino acids can be converted to the fatty acids and also stored as triacylglycerides. It usually occurs in the liver, kidney, adipose tissue and locating mammary glands. It occurs in three stages. The first stage is production of acetyl-CoA and NADPH. The second is conversion of acetyl-CoA to malonyl-CoA. The third is reaction of fatty acid synthase. Coming to the first stage, production of acetyl-CoA and NADPH. Here, the glucose is converted into pyruvate. The pyruvate from the cytosol is entered into the mitochondrial matrix, and the pyruvate present in the mitochondrial matrix is now further converted into acetyl-CoA through the PDH complex, which also involves amino acids and fatty acids. Also, the pyruvate is converted into oxaloacetate through the enzyme known as the pyruvate carboxylase. Here, the acetyl-CoA and oxalate together forms the citrate through the enzyme known as citrate synthase. The citrate present in the mitochondrial matrix is now entered to the cytosol, and the citrate over here is again formed as the oxaloacetate by the presence of the enzyme citrate lyase. Also, ADP plus PA is formed from the ATP and also acetyl-CoA from CoASH. The oxaloacetate now further formed into the malate through the enzyme known as malate dehydrogenase. Here, NADH2 is now reduced to NAD. The malate further converted into pyruvate by the enzyme known as the malic enzyme. Here, NADP is converted into NADPH2 and also carbon dioxide is released over here. The NADPH2 from the HMP shunt and carbon dioxide and acetyl-CoA is now entered into the fatty acid. Let's now discuss about the second step, which is the conversion of acetyl-CoA to malonyl-CoA. Here the acetyl-CoA is converted into malonyl-CoA by the presence of the enzyme acetyl-CoA carboxylase. Here the energetics involved were ATP is converted to ADP plus PA. The carbon dioxide is also involved over here. Let's now discuss about the third step, which is the reaction of fatty acid synthase complex. Here, fatty acid synthase along with the acetyl-CoA forms the acetyl SACP by the presence of the enzyme acetyl-CoA ACP transacylase and also releases the CoASH. Here the acetyl SACP by the transfer of the acetyl to cysteine, it forms the acetyl S enzyme. The acetyl S enzyme along with the malonyl-CoA is formed into acyl malonyl enzyme by the presence of the enzyme malonyl-CoA ACP transacylase and also releases the CoASH. Here the acyl malonyl enzyme is now further formed into beta-keto-acyl ACP by the presence of the enzyme beta-keto-acyl ACP synthase. Here the carbon dioxide is released out. The beta-keto-acyl ACP is now formed into beta-hydroxy-acyl ACP by the presence of the enzyme beta-keto-acyl ACP reductase. Here NADPH2 is now reduced to NADP+. The beta-hydroxy-acyl ACP is now further converted into trans-delta-enoyl ACP by the presence of the enzyme beta-hydroxy-acyl ACP dehydratase. Here the water molecule is released out. The trans-delta-enoyl ACP is further formed into acyl ACP by the presence of the enzyme enoyl ACP reductase. Here NADPH2 is now reduced to NADP+. The acyl ACP is now occurs of the transfer of carbon chains from ACP to cysteine and forms the acyl S enzyme. The acyl enzyme here, the second to sixth reaction occurs 6 more times and forms the product. This further forms into palmitate by the presence of the enzyme palmitoyl thioesterase. Here the water molecule also is involved. This is all about the reaction of the fatty acid synthase complex. Let's now discuss about the conclusion. The major fatty acid synthesizer in the de novo is palmitic acid, the 16 carbon saturated fatty acid. Here the main source of the carbon atoms is acetyl-CoA, whereas the source for the reducing equivalence is NADPH, whereas the source of energy is ATP. Coming to the reference, I collected this information from the biochemistry book by U. Satyanarayanan and U. Chakrapani. Thank you.